Buenas tardes. Estamos nuevamente ya conectados para continuar con la conferencia que viene. Y la que sigue es, cuyo tema se denomina Métodos Transgénicos, Cisgénicos y edición de genes en mejoramiento genético de banano para la resistencia a FOC R4T. La misma que estará a cargo de James Dye. Él es profesor en el Instituto de Entornos Futuros de la Universidad Tecnológica de Queensland, investigador por más de 30 años con intereses específicos en la biofortificación de banano, cultivo molecular para proteínas de alto valor y el desarrollo de banano modificado genéticamente resistente a patógenos. Sin más preámbulos y dando la cordial bienvenida a nuestro siguiente expositor, James Dye, dejo con ustedes a él en el uso de la palabra con su tema Métodos Transgénicos, Cisgénicos y edición de genes en mejoramiento genético de banano para la resistencia a FOC R4T. Bienvenido.
Good morning, good morning from Australia. It's 6 a.m. here, so very early in the morning and the sun's just coming up. So thank you very much for the opportunity to come and talk at this virtual conference. I think it's a fabulous idea, particularly with this current pandemic going around the world. It really has shut down so much communication, and it's fabulous to see that you have come up with an opportunity to share a lot of information around bananas, particularly at this very crucial time, because bananas are actually going through their own particular pandemic. So this morning I'm going to, or this afternoon for you, and maybe nighttime for other people, talk about methods, particularly of improving Cavendish bananas, but other bananas as well. And with very particular reference, of course, to resistance to tropical race 4. So what I would like to do, I'm going to start sharing my screen with you. And I hope that's going to come up now. Oops, where's it gone? Can we see that screen yet? Can everyone see that? Okay. So that's the topic that I'm...
Todo está correcto, todo está correcto. Podemos ver la pantalla y lo escuchamos muy bien.
Sorry, could you repeat that in English, please? My Spanish is very poor. So I'm assuming that everybody can see that. So the topic I'm going to talk about today is transgenic, cisgenic and gene editing methods for banana breeding for resistance to tropical race 4. But remembering that we'll be able to use these same technologies for other traits, other disease resistances, and other traits such as fruit quality. So if we look at what are the really huge threats to banana diseases, and I suspect that my colleague Altus Falloon may have talked a little bit about this previously in the conference, the big threat at the moment is tropical race 4 because it kills Cavendish. But not only Cavendish, it kills probably more than 60% of the different banana cultivars around the world. And it's a soil-borne fungus. It survives for more than 40 years. There's no chemical control. So it is the greatest threat to continued Cavendish production and production of many other bananas. Fusarium wilt race 1, Panama disease race 1, destroyed Gros Michel last century. But Cavendish is resistant, and one of the reasons why it took over from Gros Michel, of course. And then we have the two leaf-infecting fungi, black sigatoka and yellow sigatoka, also causing massive problems. But these are controllable, particularly by spraying. And finally, the other really big one is banana bunchy top disease. It's caused by a virus, and luckily that virus is not present in Central and South America. But it's a massive and growing problem in Africa, and very, very much an important problem in Asia as well. So what are the likely scenarios with all of those diseases? Well, tropical race 4, I believe, will certainly continue to move. Firstly in Asia, and we've seen that particularly over the last 10 to 15 years. And also in Africa. At the moment, it's only reported in Mozambique in Africa, but it's almost certain to spread. And it will probably continue to spread in South and Central America from that initial outbreak in Colombia. Spraying, of course, for controlled bacteriophaga will become less environmentally and economically acceptable, so we're going to have to think of some other solutions. And bunchy top will continue to move in Africa and potentially arrive in South and Central America. So we've got to be aware of these challenges that are going to hit us in the next decade or so. But by far the biggest disease threat is Fusarium wilt Panama disease, tropical race 4, TR4. So this is a deadly disease. It has the potential to kill more than 50% of the bananas in the world, and it's on the move, and it kills Cavendish. And you can see in the top right there, sort of classic symptoms of TR4 in the advanced stages of an infection. Around the world, we see where it's occurring, we see huge devastation in plantations. And when you cut open that pseudostem, very, very classic symptoms of vascular necrosis, that browning and blackening within the pseudostem. And in the early stages of infection, you find that just right down at the base, and then it moves up through the pseudostem. It's very, very characteristic of this disease. It's caused by Fusarium oxysporum, forma specialis cubensi. The really important thing is that this fungus can survive in soil for a very long period of time. No one really knows just how long, but certainly it's probably in excess of 50 years. And of course, it's spread in suckers on soil, on boots, vehicles, irrigation and runoff water and flood water. And in areas such as the Philippines, flooding has had a huge impact on the spread of TR4. These are some photos that I took in 2014 in the Philippines. At that time, some of the control measures were to dig out infected mats and to burn the residue in the soil. That's in the top left-hand corner. It probably has some effect, but as you see, there's just decimation. And a photo I took from the air in Davao in the Philippines, which is really pretty scary when you see those huge patches of bare ground in banana plantations, all attributable to tropical race 4. We've had it in Australia for a long time. It's probable that it moved down from Indonesia. It came into the Northern Territory, which only had a small banana business up there, small banana production, but it was quite vibrant to start with. It's virtually banana production up there is virtually wiped out. On the top left-hand side is the plantation. It's the last big commercial plantation left in the Northern Territory, and that's where we work. That's our field trial site, and you'll see there. That's an aerial photo of a planting which is probably into its first return. You'll see there's already holes in there, and if you look carefully, you'll see lots and lots of yellow plants as a consequence of TR4 infection. So quite a devastating sight to see. I sort of want the next couple of slides sort of give you an idea of this, what I've called the unstoppable movement of TR4. So if we go back to before the 1960s, it's probably TR4 was occurring in Malaysia and Indonesia. But between 1960 and about 2000, it moved quite significantly, moved into Australia, so into the Northern Territory in Australia. It moved particularly up into China, into Taiwan, etc. So you can see that it's starting to move already by the year 2000. Then 2000 to 2015 was a period of quite dramatic increase because we saw the Philippines reporting it, Oman, Jordan, Lebanon and Pakistan. But very importantly, you'll see there the first outbreak in Africa, and that's down in Mozambique. Then we go to 2018, so not much later, and now we see that Israel's reported it, Vietnam's reported it, Laos, Myanmar, India, and interestingly in the UK, not a big banana producer. And then 2019, a lot more movement, but also very importantly into Colombia and also in Turkey. So really quite scary when if we just go back and just look at that very quickly, you'll see that it just keeps moving. And maybe by the end of this decade, we're going to see a lot more movement, probably in South and Central America, but also in Africa. It's likely to move a little bit less quickly because we are very, very aware of the disease. We're very aware of the fungus and how it moves. And so we're probably going to be able to slow it down a little bit, but we're not going to be able to stop it, I don't believe. And so that's just a list of the countries and the dates. And 2020 was Coromos, and we'll probably have some more additions to that. It'd be unlikely that we're going to see this disease stop moving. So I think the really important thing, so surviving Panama disease, TR4, we've got to really think strategically about this. And I think very importantly, once TR4 reaches a region, spread is almost inevitable. And so we really have to have a mid to long-term solution to the TR4 problem. And in reality, that mid to long-term solution is going to be resistance. And we do have a lot of different strategies that we can do. We've got options. And I put down the four major options that are available to us at the present time. And one is tolerance selections, and certainly we've heard a lot about GCTCV218. It's being developed out of Taiwan and used widely in the Philippines. And that's a good example of a tolerance selection. It's a Cavendish type with apparent tolerance. There are a number of breeding programs, conventional breeding programs around the world, and some of those have already resulted in the development of TR4 resistant cultivars. The important thing to remember here is that conventional breeding will end up with a new cultivar. It won't be Cavendish, it'll be something different. And then more recently, and we can probably call them the new breeding technologies of genetic modification, and much more recently gene editing. And the result here is that you can actually edit the cultivar or modify the cultivar that you're actually growing. So mid to long-term solution, definitely new or improved cultivars. So the reality of the future of export bananas, I believe, is either a conventionally bred new banana cultivar, which is going to be different to Cavendish, but with disease resistances, high yielding, transportability, and excellent taste and texture. So for an export banana, it's got to have all of those characteristics. Or a genetically modified banana or gene edited Cavendish, but with multiple disease resistances. So you're already starting with a cultivar that is high yielding, it transports well, and it has excellent taste and texture. So you've just got to add disease resistances to that. I thought I'd just mention very quickly the difference between genetic modification and gene editing, because sometimes these get a little bit confused. So genetic modification, and I'll include in here genetic engineering, transgenics, and cisgenics. It's essentially the addition of new DNA into a plant through transformation that almost always includes foreign DNA. So it almost always includes DNA other than banana DNA. And it is this concept of adding new DNA. Gene editing, on the other hand, is a very, very precise technology, and it's targeted modification of the plant's own DNA. So we're not adding anything, we're actually just going in and modifying what's already there, including deletions, substitutions, but also you can go right through to addition of entirely new genes. So for non-GM editing, and this is, we'll sort of talk about this much more towards the end of the talk, there must be no foreign DNA, no new foreign DNA added. So this is very important. Only banana DNA. If we're adding an entire gene, it has to be just a banana gene. But in the simplest forms with gene editing, we're doing deletions and substitutions, and that as long as there's no foreign DNA, that's considered non-GM editing. I should mention that only 2 days ago, the two of the scientists who developed the most common form of gene editing, and that's the CRISPR technology, won the Nobel Prize for chemistry. And so that was just to give you some idea of just how important this technology is and how it's changing so many different facets of biology in the world. Cisgénics, just to reiterate that, cisgénics is the transfer of a complete banana, in bananas this is, of a complete banana gene from one banana to another, but it is by transformation. An example would be the FOC race1 resistance gene that obviously occurs in Cavendish, moving that to Gros-Michel. So that would be to give Gros-Michel resistance to race1. So that would be a cisgenic. But usually that transfer also includes a small amount of foreign, and that is non-banana DNA. And therefore it's classified as a type of genetic modification, type of GM. It is much more acceptable, but it is still classified as GM, and when it's classified as GM, it carries with it a number of restrictions, particularly around regulation. So if we look at all of the technologies that we can use, and that includes breeding, and that's conventional breeding, natural mutations, they're things like the somoclonal variants, and also mutagenesis, that can be either chemical or irradiation mutagenesis. Those are relatively old technologies. We've got transgenics, which include cisgenics, that have been more recent. And then in the middle there, this is the gene editing technologies, which are divided into the very early stage or simple technologies called SDN1 for site-directed nuclease 1, right through, and so they're just deletions and substitutions in a DNA, right through to adding complete genes, which is called SDN3. It all comes under the gene editing banner. So that's just a slide showing you really all of the different methodologies that can be used. Now, when we develop new cultivars, new varieties, there's various pathways. So selection, conventional breeding, and somoclonal variation is not regulated at all, and is not required in labelling. Chemical mutagenesis or irradiation is usually not regulated and is usually not labelled. Quite recently, however, in France, the courts recognise chemical mutagenesis and irradiation as really a form of genetic modification. So we're not quite sure where that's going. That's particularly in the European Union. Not a lot more has been said about that, but we just have to keep that in mind. So genetic modification is regulated, and in many instances it's labelled. For gene editing, at the moment, because it's a relatively new technology, not all geographic regions have decided how they're going to go with this. It's mostly not regulated, but there is geographic variation and there's definitional variation, and we'll talk about that. If it's not regulated, it's not labelled. So gene editing is getting towards the same level of regulation as conventional breeding, which is really very, very beneficial. So where do we get these genes from? And this is really important. For somoclonal variation, that's that's within the banana cultivar itself. Really, we're just trying to select out from that cultivar something that has a little bit more tolerance. For conventional breeding, the genes can be accessed from what's called the accessible gene pool. So from bananas, obviously, that can be crossed one with another. With genetic modification, there are really no limitations on where you can get genes from, and you can actually generate whole new genes. Gene editing is within bananas itself. So this is altering genes that already occur within bananas, or moving entire genes from one banana to another. So let me talk about our programs, because this gives you a pretty good idea of the sorts of things that can be achieved through both modification and genome editing, gene editing. So we've been working with TR4 for a long time. In fact, we began our TR4 resistance program back in 2000, so it's 20 years ago. As I sometimes say, we started working on TR4 before it became a superstar disease. It certainly is that now. We went looking for resistance genes, and we went looking for resistance genes in the wild germplasm, and particularly in the diploid bananas. As most of you are aware, bananas, particularly acuminata, evolved in Southeast Asia primarily, right down into the tip of Australia. And so right throughout that region, we can find diploid bananas, and some of those have resistance to all sorts of diseases, particularly in this instance to TR4. And we isolated a gene which we called RGA2. It's a resistance gene, and it's from a diploid banana called Musa acuminata malacensis. In 2006, we commenced our genetic modification of Cavendish. We had a couple of different genes, and we moved that gene around for resistance to TR4 in Cavendish and race 1 resistance in ladyfinger. So by 2011, we demonstrated we had race 1 resistant ladyfinger. In 2012, we started our field trials in Darwin with Cavendish, and the first stage was completed in 2015. 2017, we commenced gene editing of Cavendish for TR4 resistance, and then we started our second phase 2 TR4 Cavendish field trial in Darwin in 2018. And I'm going to talk particularly about a number of those instances. So that phase 1, that resistance gene identification. So on the left-hand side, there is a small seeded banana. This is Musa acuminata malacensis. Absolutely stuffed full of seed, and those of you that have seen banana seeds, they're very, very tough little seeds, and you certainly don't want to bite on them. We recognize that in this particular malacensis, there was a single dominant gene that controlled TR4 resistance, and so if the gene was present, you got essential immunity. And if the gene was absent, then you got susceptibility. And so we were pretty excited about that. Single dominant genes for resistance are really very, very good to work with. So we identified this resistance gene primarily by genomic analysis and expression in resistant versus susceptible siblings. And essentially we had a group of malacensis, some of which are resistant and some of which were susceptible. And so we were able to, by comparison, pick out genes that appeared to have influence on that resistance. So we transferred genes to Cavendish and planted those in field trials in Darwin. So what we'd essentially done is we'd taken this single resistance gene, RGA2, from a wild banana and put it into Cavendish, and the Cavendish we put it into was Grand Name. So the next part of that was the initial field trials starting in 2012. So this is a map of Australia. Here we are in Brisbane, and that's where I am at the moment. So this is essentially in the subtropics. So here we have a banana industry down here. It's not very big. Our major banana industry in Australia is around the Tully Valley, where TR4 does occur, but only in a relatively small number of plantations, even though that's increasing over time. This is in the Northern Territory up here. The vast majority of it is sparsely populated. And as you can see there, there's a desert, the Tanami Desert. But up here, this is in the wet tropics. And our field trials are a place called Lambles Lagoon, which is about an hour's drive out of Darwin, which is the state capital or the territory capital. Let me tell you a little bit about Darwin Fruit Farm, and that's where our field trial site is. It is a large commercial plantation, which is very severely affected by TR4 and has been for more than 10 years. They continue to grow bananas there because they supply the local market. It's the only plantation that has sufficient volume to supply the local market in the Northern Territory. And usually it's a plant, the plant crop, the first return and maybe the second return are harvested before that site has to be ploughed in because of the effect of TR4. Our field trial site is adjacent to these commercial plantings. The important part about this is that we have a complete history of every plant in the trial, which we think is probably a unique set of data that we can say, okay, this is the plant was planted in 2012 and this is what happened to it all the way through those 3 years. All infections are confirmed by PCR and in many instances also by sequencing. So we've got invaluable data such as rate of infection, spread, recovery, and specimen collections. So that's an aerial photo of our field trial site. This is where the planting is. This is a 10-hectare space here. We've got large screen house here where we take the plants up from Brisbane and they're acclimatized. And we've also got a small lab here where we can do DNA extractions, we can grow up TR4, etc. So it's actually a very, very well set up area for doing TR4 resistance screening. So the trial, as I said, was planted in 2012. It ran for 3 years. The reason we stopped after 3 years was another disease, Cavendish freckle, was found in the Northern Territory. And to eradicate that, they had to eradicate all bananas in the region, and that included our field trial. That was the only reason we stopped that. The controls we had was the GCTCV218. That's the tolerance selection from Taiwan that's very popular in the Philippines. DPM25, which was developed in Australia for resistance to subtropical racefall, not tropical racefall. And we also have Grand Nain and Williams as controls. All the transgenic lines were in Grand Nain and they were transformed with a single banana resistance gene. So this is the result after 3 years. There's some important information here. So these are the controls. These are the controls in blue. So the most susceptible was this DPM25. So after 3 years, 100% of the plants were infected. Around about 87% of the plants, the Grand Nain plants, were infected. The interesting thing is that Williams appeared to be more tolerant. It was about 66% compared to Grand Nain. And 218 was around about the same level of susceptibility as Williams. There wasn't statistically any difference between them. And here are the 7 transgenic lines we took up. And they all had the RGA2 gene in them. So 4 of those lines had significant levels of resistance, and 1 of them appeared to be totally immune. So no infected plants after 3 years. So here it was about a 14% infection, and these two were about 20% infection. But I should stress here, it was only a small trial. this was really our initial trial to see whether the gene provided any true resistance. And this is the progression, and it's really quite interesting. So again, the reds here are the controls, and you'll see here, this is DPM25 here, and this is grandnane. And you can see a very steady increase in the number of plants infected over time. Even though over the first 12 months, you don't actually see a huge amount of infection. And it's very important. I think often we see field trials of 12 months, maybe up to 18 months for TR4 resistance, nowhere near long enough. Really, you need trials of 3 to 5 years, or maybe at least 2 years to really get an idea of the level of resistance. So for instance, with Williams, we didn't get any infected plants in 218 for about 12 months after planting. So if your trial went for 12 months, you'd say, oh, 218 and Williams are resistant. Go for 3 years and bingo, you're up around about 67% infection. The green lines are the transgenic lines, and this RGA23 here right down the bottom with no plants. And then we have ones like these two, RGA22 and RGA24, which for 2.5 years didn't have any infected plants and then had a few infected plants right at the end. So they'd be probably very commercially acceptable cultivars. I took this photo the day that we had to destroy the trial, so every single plant was cut down and assayed. And we ended up with 1 highly resistant and 3 resistant lines. And when you cut that open, this is the RGA23 line, absolutely clean pseudostem as compared to the pseudostem of infected control. One of the really important things we found is that this is a southern analysis, a southern gel, is that grandnane contains the RGA2 gene. So the question is, if it contains the RGA2 gene, why is it not resistant? And the reason for that, and this is what's called the expression levels of the RGA2 gene. This is grandnane here, and this is RGA27, which was one of our lines that was susceptible. You'll see that the gene essentially isn't working. It's not producing anything. However, the resistant lines from their RGA2 gene produce a huge amount of messenger RNA, which is the intermediate between the gene and finally the promoter, the protein that provides that resistance. So even though grandnane has that RGA2 resistance gene, it doesn't work. And that's quite important. We've published that, so this was published in 2017 in quite a very reputable journal, Nature Communications. So then we went to phase 3, and this was the expanded field trials to identify lines that we can take right through to deregulation. So this is taking those field trial lines or those resistant lines all the way through the process so that they can be sold in the open market. So what we've done is we've taken, this is a much, much larger field trial. Large numbers of RGA23 and the other 3 lines, other 3 lines. So we took 4 lines, all the resistant ones, into a much larger field trial. We developed new RGA2 lines, both in grandnane, but also in Williams. So the initial ones were in grandnane. So now we've got lines in Williams. We're also looking at resistance genes from other sources. And we planted this second trial early in 2018. Again, showing that map, here we are down here, Lambles Lagoon. So we're at exactly the same spot as we were for that initial field trial. So this is a trial up the top. As you can see, this is a big field trial. We wanted to make sure that what we saw on a limited scale was also applicable to very large scale field trialing. And on the left and the right hand side there, you'll see two of the transgenic lines, and their bunches, their good bunches, look exactly the same. These photos were taken very recently, September 2020. One of the challenges of COVID-19 is that we've been very limited in our ability to travel from Brisbane up to our field trial, but we were able to go up in September, and so we took these photos. This one is an earlier photo. We've had sequential plantings of the different lines. The original lines were planted first, and then some of the new lines and some of the new resistance genes. It's so this is a large field trial. It's going to run probably for 5 years. It's a randomized block design. All of the plants were transferred to Darwin Fruit Farm in vitro and acclimatized for 3 months. 4 original ground name RGO2 lines, the results are almost identical to the phase 1 trial. And we're collecting yield, cycle time, and the long-term history of every plant, and we will ultimately publish those results. So I'll just give you some photos of those trials. These are controls. These ones were taken in 2019, so this is 12 months, a bit over 12 months after they're planted. They're planted in March, and you'll see early symptoms, and when you cut open the pseudostem, you get that vascular discoloration. And so these are very, very typical symptoms. These are more advanced infections with much more vascular browning. And these are all, of course, non-GM controls I'm showing you here. And the sort of dark brown pitting right in the middle of that pseudostem. So what we do is we've now developed specific tests for tropical race 4, subtropical race 4, and race 1. And these are sets of primers which differentiate between each one of those very simply. And we do all of our DNA extractions on site, and then we bring all the samples, all those DNA samples back to Brisbane, and they're screened by PCR, but they're also screened, many of them, by sequencing, and sometimes of the PCR product. But for those interested, we also clone that and sequence it as well. To give you some idea around those PCR results, this is a very, very early stage. So you're just starting to see symptoms of TR4 coming up, just one leaf. And when you cut that open, if you cut it right at the base, you're starting to see that vascular discoloration. And you go up into the mid-pseudostem, and you're seeing a little bit more vascular discoloration. At the top, tiny bit, yeah, not very much at all. Nothing in the peduncle, and of course, nothing in the fruit. So when we go and do those PCRs, this is PCR positive, this is PCR positive, this is PCR positive, but these are negative, as we would expect. When we go to a sort of mid-stage infection, and this is a plant, this is the plant that we used at the mid-stage infection, and all of a sudden you're seeing a lot more vascular discoloration at the base and mid-stem, and also up in the top. And you do see these little necrotic flecks in the peduncle, and sometimes you'll see little necrotic flecks in the fruit as well. And we were particularly interested to know what this was about. But then when you go and do the PCR, PCR positive, PCR positive, PCR positive, as we found with the early stage infection, nothing out of the peduncle and nothing out of the fruit. So the fungus is not present. Even though you'll get these little brown flecks, the fungus isn't present there. And then we can go to an advanced stage infection. So this plant would probably be dead in the next couple of weeks, and all the leaves are infected. This is a severe infection. When you cut it open, huge amounts of necrosis at the base and also mid-stem and also at the top. And this has a very, very characteristic smell, of course. And again, you see these little brown flecks. We collect these and sometimes in the fruit. But again, exactly the same result. We never, ever are able to identify TR4 either in the peduncle or in the fruit. And that's a very, very important and very good result. And we can do that over and over again, get exactly the same result every time. Okay, so let's go to the results from that second field trial. And here after 12 months, and this is why we always need to look at 12 months rather than 3 years rather than just 12 months. So that's our control, 16% infection after 12 months. In the 4 lines, 3 of them had a very low level of infection, 2%. 1 line didn't have any. Much more importantly, when we go to 2.5 years, and that's September this year, Grand Nain is up to 52%. And we'll probably see that dramatically increase over the next 6 months. 1 line had up to 14% and pretty much the same result as we had in the first field trial. 1 line, no plants infected. 1 line with very small amount of infection, and 1 line with a small amount of infection. So these ones, this one, is it immune? Don't know. Always be careful to say that. A very, very high level of resistance, as is this one. These two lines have very, very high levels of resistance, and this has very good resistance. So the interesting thing, and what we've done, is to extrapolate from those numbers of plants in the field and their yield to give us approximate yield per hectare from each one of those lines. And we work on planting of around about 1,600 plants per hectare. And when you do that, this is the infection level here, these are the lines, infection level, and an extrapolated yield per hectare. So the important thing is that we would end up with around about 25 tonnes per hectare from the Grand Nain control. This is in the second return. And our best line, we're more than double that. So nearly 54 tonnes per hectare. This one is an interesting line because even though it has 14% infection, it actually has a higher yield per plant than the Grand Nain control, even though we didn't do anything to increase the yield. So this is a really important result for us. So we've got 1 line that has no infection and is yielding extremely well on a per hectare basis. So what we can say is that Cavendish bananas with immunity to TR4 are a reality. So these are available. And that's very important. So certainly Cavendish will not disappear. However, technology always moves on, and this is very important. So now what we're looking at is to develop that same level of resistance, but through gene editing of Cavendish and of course other bananas. And going back to that slide I had previously, with the SDN1, SDN2, SDN3 in that center there, these are the gene editing technologies. The reality of gene editing in bananas, okay. So really we're looking at either knockouts, so knocking a gene out, substitution, so changing a couple of the nucleotides, maybe changing how genes are expressed, or insertions, and that is putting in a completely new, or not new, but putting in a whole banana gene. And very importantly, we can't have a selectable marker, except in banana breeding lines, but that's a small area. So it's no selectable marker, and that's quite important. We have to have transient expression, and we use CRISPR, and we have to have transient expression of the components of CRISPR, which is a Cas9 protein and a guide RNA. So we have to do that in various ways. The immediate targets, the ones that we're looking at immediately, are PPO, so this browning. Is it possible to develop non-browning bananas? And yes, we believe it is. To potentially manipulate ethylene, the amount of ethylene, and therefore control ripening. But most importantly, disease resistance, and the one we're focused on right now is, of course, TR4. And there are multiple gene editing technologies. There's CRISPR, TALENs, meganucleases, and zinc fingers. This is the one, CRISPR is the one that we're using, and we have a license to use that technology. It's by far the most applicable technology, we believe, to bananas, because it's a research tool as well as a developmental tool. And this is just to give you an idea, and you don't have to remember any of this, but essentially what it is it's a pair of scissors which can cut DNA at a very, very particular place. And we can define that where that cut occurs. And so we can cut a little bit of DNA out. And this is called non-homologous end joining. And this is the most common usage of CRISPR in plants at the moment, but also homology-dependent recombination. So this is where we would add an entire new banana gene, resistance gene. So that's how it works. There are a number of components. We need the technology. We're using CRISPR. We need ability to edit and with markerless regeneration, and that's we use embryogenic cells, etc. We have to have an understand the genes, a donor genome and a recipient genome, in this case, grand name, and then our editing strategy. We've demonstrated already that CRISPR works in bananas, and we published this, that when you knock out this particular gene, phyto-N desaturase, you produce bananas that are albinos. And we've done that, and they're the little albino bananas there. So we know that CRISPR works very, very well in bananas, and we've published that already. So where we're going now is to use protoplasts, and these are single cells. So we take these cells here. This is what we normally use for transformation. Taking these cells, removing their cell walls, so you get absolutely single cells. And so we'll be able to edit, or are in the process of editing, these single cells. But the big challenge was to take one of those and get it to develop into an embryo, and then finally to develop into plants. So from one of those single protoplasts, it's possible to go from here all the way through the process to make protoplasts, into developing embryos, and into plantlets. So we know we can do that now. So the process now, of course, and we can, is to use these for editing, and those are in the process of coming through. So the strategies, what can we do? So for TR4 resistance, we can upregulate resistance genes, and that comes under that SDN1 terminology, and that's what we're doing first up now. The other way is to identify what's known as susceptibility genes. And it seems strange that a plant would have genes that make it susceptible to diseases, but these genes are often involved in important processes in the plant. So at the moment, we're identifying some of these susceptibility genes and then knocking them out. So instead of putting a resistance gene in or making a resistance gene work better, we're knocking out a susceptibility gene to generate resistance. So that's a second strategy. Third strategy, which is more challenging, is to take a whole resistance gene, for instance, the resistance gene from Mucor acuminata malacensis, and actually put that whole gene, back into cavities. This is this SDN3 technology. This is very, very difficult to do. It's not very efficient, and also SDN3 isn't necessarily recognised throughout the world as applicable for non-regulation. But we do need a very detailed knowledge of both the Cavendish genome and their wild relatives. And that's what we've been doing. And we've got Cavendish, our Malacensis resistant and our Malacensis susceptible. And these ones, so for instance, Cavendish is resistant to race 1, but susceptible to most other things. This one has resistance genes for race 1, tropical race 4, but also black and yellow Sigatoka. So we're very interested in this for the future. And so we've sequenced both of these. We have very, very detailed sequence that gives us the opportunity to target very precisely the genes that we want to go after. So it's a rapidly changing landscape. I think importantly, the Colombia outbreak has brought the reality of the TR4 threat to the Americas. So, and just like COVID-19 has changed our social life, tropical race 4 has changed our banana life. And I think it has changed forever, and we're going to have to be much more open to the various ways that we're going to cope with this new challenge. So really the midterm options, I think, are going to be a new and different export banana by conventional breeding. So this would be something else other than Cavendish. A genetically modified Cavendish, which is already available. A gene-edited non-GM Cavendish, which we think is certainly in the pipeline. Maybe even going back and developing Gros Michel with resistance to race 1 and tropical race 4. And maybe also doing the same sorts of things with plantains, which are very, very important, particularly in Ecuador. So GM is politically challenging, but it's acceptable in the US, Canada, Australia, Japan, Philippines, probably Colombia, and I'm not sure about some of these other countries. Gene editing, non-GM gene editing is now accepted in many countries and is being seriously considered in others. So this is a much more acceptable technology. And I'm going to finish with this slide. And this is a slide which gives you an idea of where gene editing, this is non-GM gene editing, is acceptable. So in North America, so both Canada and the United States, acceptable as non-GM. Colombia, Brazil, Argentina, Chile, there's a case-by-case basis, but if there's no foreign DNA, then it's not regulated as a GMO. Australia's in that same position, and so is Japan. So these are the areas that have moved most quickly. Israel also is not regulating gene-edited plants or food. The ones in yellow are where the discussion is going on, and this is, and in most instances, it's probably going to say, yeah, when gene editing is non-GM. Russia is almost certain to go down that path as well. United Kingdom is probably going to, because it's leaving the UK, is probably going to go down that path as well. So it's looking increasingly likely that gene editing is going to be the most applicable non-conventional plant breeding way to develop the next generation of bananas, particularly the next generation of Cavendish. So QUT, we've already developed TR4 resistance in Cavendish, and these are in advanced field trials. We're developing gene editing technologies for bananas. CRISPR works in Cavendish. Protobios technology is progressing. We've got the genomes, and the knockouts are progressing. We've got Gros Michel and plantains in the pipeline as well. So thank you very much for the opportunity to talk to you all this morning, Australian time, and whatever time it may be, wherever you are. These are the groups that have, this is my team at QUT, and we've been supported by many others in developing this technology. So thank you very much.
Muchas gracias. Gracias a James Dye por su extraordinaria conferencia sobre este tema tan actual, métodos transgénicos, cisgénicos y edición de genes en mejoramiento genético de banano para la resistencia FOC R4T. Gracias. No sé si haya alguna pregunta para el expositor. Pueden conducirlas y dejarlas en el chat si hay alguna interrogante. De momento no vemos ninguna. No obstante, entendemos que ha habido, sin lugar a dudas, una gran acogida por parte de quienes se encuentran conectados con nosotros en esta plataforma virtual. James, no existiendo interrogantes de momento, agradezco a nombre de AEB por vuestra extraordinaria y brillante participación y lo comprometemos para que en nuestro próximo evento, el año venidero, Dios mediante, también nos acompañe. Gracias y una buena tarde.
Thanks very much.
Importante indicar que de esta manera y con esta conferencia estamos dando punto final y el cierre de el 17ª Convención Internacional de Banano 2020. Un evento que ha tenido una aceptación extraordinaria, en realidad ha rebasado todas nuestras expectativas y entendemos que esto guarda relación, sin lugar a dudas, en virtud de la calidad de cada una de las ponencias, de las brillantes intervenciones de cada uno de los expositores, tanto en conferencias individuales como en mesas redondas. Así que queremos hacer extensivo, a nombre de la Asociación de Exportadores de Banano del Ecuador, nuestro agradecimiento a los más de 2.000 participantes que estuvieron junto a nosotros desde el primer día, desde el día lunes 5, en que fue la inauguración formal de la 17ª Convención Internacional de Banano, y posteriormente a ello, desde el 6 hasta hoy, 8 de octubre, de cada una de las conferencias. Es por ello que hemos decidido, a nombre de la asociación, su directiva y cada uno de sus socios, hacer extensiva una cordial invitación a todos y cada uno de los participantes, de quienes se inscribieron, de quienes confiaron en nosotros, para darles esta buena noticia. Cada una de las conferencias, así como nuestra feria virtual, estarán en la plataforma hasta el día viernes 16 de octubre. Así que si usted quiere tomar algún apunte adicional, si usted quiere volver a ver porque quiere conectarse con algún dato en específico que le gustaría replicar, cada una de las ponencias van a estar en la plataforma virtual, así como recorrer la muestra comercial, repito, hasta el viernes 16 de octubre. Gracias, gracias, porque esto solamente evidencia la confianza que tienen en este trabajo de equipo que realiza la Asociación de Exportadores de Banano del Ecuador, su directiva y, por supuesto, su director ejecutivo, el ingeniero José Antonio Hidalgo Molina. Queremos aprovechar también la oportunidad para poder hacer extensivo un reconocimiento de gratitud a nombre de quienes conformamos el clúster bananero del Ecuador, de cada uno de sus integrantes, que no habría sido posible esta unidad de no ser posible por el apoyo de ASOCAR. Y quiero, ahí está en pantalla, este agradecimiento de quienes conforman el clúster bananero del Ecuador, que dice lo siguiente: considerando la valiosa colaboración con el sector bananero ante los desafíos que enfrenta nuestra industria, queremos exteriorizar nuestro agradecimiento a ASOCAR y cada uno de sus integrantes: Papelera Nacional, Grupasa, Procarsa, Carcopel, Incarpal, Industria Cartonera Ecuatoriana. Acuerda expresar un justo agradecimiento y dejar constancia de la relevancia para el clúster bananero ecuatoriano de vuestra colaboración y esfuerzo, porque sin la ayuda de cada uno de los integrantes de ASOCAR no habría sido posible la conformación, la unidad, la compactación de este clúster bananero ecuatoriano. Así es, expiden este acuerdo este acuerdo de agradecimiento, este presente testimonio de este merecido reconocimiento, dado y firmado a los 8 días de octubre del 2020 en la ciudad de Guayaquil por el ingeniero José Antonio Hidalgo Molina, director ejecutivo de AEBE, el ingeniero Richard Salazar Veloz, director ejecutivo de Acorbanet, el ingeniero Leonidas Estrada Vázquez, presidente de Agroban, el ingeniero Clever Sigüenza Rojas, de la Comisión de Banano de la Cámara de Agricultura de la Segunda Zona. Y por supuesto, también firma quien es el vocero del clúster bananero ecuatoriano, el ingeniero Juan José Pons. Reiteramos nuestro agradecimiento a Azocar. Adicionalmente, queremos también exteriorizar a nombre del clúster bananero ecuatoriano nuestra gratitud al Banco Guayaquil. Así es, el clúster bananero Ecuador, considerando la valiosa colaboración con el sector bananero ante los desafíos que enfrenta nuestra industria, expresamos nuestro agradecimiento al economista Angelo Caputi, presidente ejecutivo del Banco de Guayaquil. Acuerda expresar un justo agradecimiento y dejar constancia de la relevancia que tiene el apoyo del Banco Guayaquil para la conformación e integración del clúster bananero ecuatoriano. Gracias al Banco Guayaquil por su cooperación y esfuerzo. Es por ello que exteriorizamos a través de este reconocimiento nuestra gratitud. Lo firman el ingeniero José Antonio Hidalgo, director ejecutivo de AEB, el ingeniero Richard Salazar, director ejecutivo de Acorbanet, el ingeniero Leonidas Estrada, presidente de Agroban, el ingeniero Clever Sigüenza, de la Comisión de Banano de la Cámara de Agricultura de la Segunda Zona, y por supuesto también agradece por este apoyo tan importante del Banco Guayaquil el ingeniero Juan José Pons Arízaga a nombre del clúster bananero del Ecuador. De esta manera culminamos esta actividad con estos reconocimientos muy merecidos, por cierto, y nuestro agradecimiento, reitero, a todos quienes se sumaron y participaron de manera activa en cada una de las conferencias que estarán a vuestra disposición en la plataforma virtual hasta el viernes 16 de octubre. La muestra comercial, nuestra feria y también cada una de las conferencias. A nombre de AEBE, Asociación de Exportadores de Banano del Ecuador, y del CIBE, el Centro de Investigaciones Biotecnológicas del Ecuador adscrito a la ESPOL, les agradecemos el haber sido parte de la 17ª Convención Internacional de Banano 2020. Nuestra gratitud imperecedera. Quedan a vuestra consideración las conferencias y la muestra comercial hasta el viernes 16 de octubre en virtud del éxito arrollador y extraordinario que ha tenido, para que ustedes puedan acceder a cada una de ellas. Si Dios lo permite, volveremos a encontrarnos en el la 18ª convención, si Dios lo permite, el año venidero. Un abrazo fuerte y que tengan una buena tarde.